Welcome to this review video over Lesson 3.1, and Lesson 3.1 is about how we detect cancer. And before we begin with the details of this lesson, I want to give a shout out to Chad Shelley, fellow PLTW biomed teacher, who created this PowerPoint. What are the fundamental characteristics that all cancers have in common? Well, first and foremost, cancer is a disease in which the DNA has become damaged, so it has a genetic component. Now, what causes these genetic components? Could be that you just simply inherited the genes. But more often, there's an environmental component, such as smoking, exposure to radiation, including UV light, your diet, your overall health status, lack of exercise. All of those can lead to damage to the DNA, which will cause cancer. Now, if you catch your cancer early and you get it removed before it can spread, the cancer may not cause too many problems, and in fact, it can be controlled and you potentially may be cured. But if it spreads, and the fancy term for spreading is metastasizing or metastasis, process of spreading, there can be some serious potential dangers that potentially could be fatal. Now, as you get older, the chances of you having cancer become much more probable, because over your lifetime, as you've walked through life, you've picked up mutations. There's a natural mutation rates in humans, plus you throw in all of the environmental factors from a modern society that could lead to a buildup of these mutations, and eventually you get enough of them, you could get cancer. Now, the other thing you want to know, cancer can affect any part of your body. So there's not a single part of your body that cannot get cancer. All right. There's two main genes that are involved with cancer, and the first one we're going to talk about here are the proto-oncogenes. Now, proto-oncogenes control cell division by telling the cell to begin to divide. So kind of think of these potentially as growth factors. Now, cancer can be caused when these proto-oncogenes are turned on too much. In other words, the cells are getting too much of a signal to divide, and therefore they're going to divide too much. Now, when they're in the proto stage, or the proto-oncogenes, they are functioning normal. Now, a mutation into these proto-oncogenes could cause this gene to change into a full-blown oncogene, and this is the gene that's being expressed out of control that leads to too much cell division and therefore too much cell growth. Now, how can we get these mutations? Simply could be those environmental factors, or you could have inherited it. But there could also be some other genetic problems that can lead to this, such as a translocation. A translocation of the promoter can cause this oncogene to be turned on more than it should be, and that will lead to the uncontrolled cell growth. Now, there also could be a mutation where you basically have a multiplication or a duplication of these proto-oncogenes, which can lead to them being expressed way too often, and therefore an increased amount of cell division, therefore cancer. Now, the other type of gene that is involved in cancer is called a tumor suppressor gene. These are the genes that are designed to be a normal quality control when it comes to cell division. And when they're functioning correctly, any mutations that develop during the replication process will either be fixed, or the cell can go into a series of self-destruct mechanisms so that this kind of cell cannot proliferate. Now, tumor suppressor genes will normally be able to stop cell division at different points, and they'll use this to do a kind of a spell checking or quality control stuff. The most famous of these is the p53 gene, right? And this is known as the guardian of the genome. Now, many of its functions, probably its most important is it's going to trigger apoptosis, which is programmed cell death. This is when there's too many mutations that cannot be repaired, and p53 says, I just can't fix this one, so we got to get rid of it, and that'll cause apoptosis. But if they haven't reached that threshold of damage, then p53 can actually activate the enzymes that will repair the DNA. All right? It can also halt the cell cycle until all these repairs have been made. All right. Now, mutations to p53 or any of the tumor suppressor genes will lead to cancer because you don't have these natural checks on any kind of damage to the DNA. So your proto-oncogenes will trigger cell growth, and then your proto-oncogenes can become oncogenes, and that will become out of control cell growth, and the tumor suppressor genes work in the opposite progression in order to keep cell division from getting out of control. All right. Cancer cells they're kind of different. Because they're out of control, they're not affected by that Hayflick limit that you learned about probably in your Bio 1 class. These can proliferate in a laboratory situation indefinitely. When we were looking at our different cancer cells that we bought from CellServe, we had those LMR 90 cells. Those were from a mouse that died in 1948, and we've been able to keep them alive ever since. HeLa cells, late 50s, early 60s, same situation. So as long as you are giving these guys proper nutrients and a good environment, they can potentially live forever in a petri dish. Right? They're not going to exhibit that contact inhibition. Normally when cells are being touched on all sides, they're happy and they'll stop growing. Cancer cells, they don't pick up that signal and they just keep growing. You'll get that big lump on the petri dish, and that's your tumor. All right. They also undergo the morphological changes. Now, in our class, we took those wonderful pictures with our iPhones, and then we use a kind of a software app where we could put two pictures next to each other, and you could really see the difference between the normal cell and the cancerous cells and how different their shape was. So remember, morphological just means shape. Cancer cells can grow even when the environment's not great inside your body. So they're under less strict environmental conditions. So they're just not quite normal, so they can do a lot of crazy things. But none of those contribute to maintaining your own homeostasis. All right. Now, another thing we notice is that cancer cells can have an abnormal number of chromosomes. This includes basically aneuploidy, which means they're going to have not a multiple of 23 in humans, but they'll have a few chromosomes. So maybe we're supposed to have 46, maybe we got 54, 67, some crazy number like that. And these chromosomes will have a weird shape to them also. Diagnostic imaging, which was a big part of this lesson, and as you see here on your screen, these are the four main ones. Now, an X-ray is probably the one that we're most familiar with because we've probably have all turned an ankle or broken a bone or something like that, and we've gotten an X-ray that will clearly show the broken bone or anything like that. But these are also good for lung diseases. You can pick up tuberculosis looking at a lung X-ray, and you can certainly pick up any tumors in the lungs using that. Now, in your digestive system, you can also get an X-ray, but you've got to take basically some kind of fluid that you're going to drink that contains a metal. Particularly, it's usually iodine or it's going to be barium. And this will allow the X-rays to kind of be absorbed, and you can see the message or see the image. So those kind of pictures of your digestive tract are really, really neat to see, because some of them can be filmed live and you can see the peristaltic action of your digestive system. CT scan and MRI, they're kind of similar because they're both designed to show the soft tissues. A CT scan is going to use an X-ray, and what it's going to do is going to take cross-section. So imagine your body is like a piece of bread or a loaf of bread, and we're taking slices of it as a picture. Then this is going to allow us to see the internal organs and structures a lot easier. So remember, a CT scan is going to use an X-ray, but an MRI is going to use magnets. And what happens is it creates a large magnetic field around you, and you have certain ions in your body that will emit a radio wave, and the MRI machine is going to pick up those radio waves and make an image out of it. And so the machines kind of look the same and they kind of do the same thing. They just use a different method to do it. Both of them are used to see soft tissues. All right, bone scan. This would be something that Mike Smith would get in our little story. You're going to basically take in a radioactive tracer that's going to be absorbed by your bone, and then you're going to be able to take, it's not quite an X-ray image, but it's going to be something similar, and you're going to be able to see where that radioactive tracer has been absorbed by the bone and where it has not. And based upon the view that you get from there, you'll be able to tell if it's cancerous or it's bone growth or any kind of picture like that. So X-rays, CT scans, MRI, and bone scans are the major ways to do diagnostic imaging when it comes to cancer, and of course other medical maladies. All right, DNA microarrays. This is kind of the coolest thing when it comes to this lesson, because we're using molecular technology and you really got to find out which exact genes are going on. So the fact that you're using biotech and computers, to me, this stuff's just really, really cool. All right, so what the heck is a DNA microarray? Well, DNA microarrays measure the number and amount of genes turned on and off in cancer cells versus normal cells. So, of course, you're going to use those to figure out which cells are you going to see in a normal cell and then which cells you're going to see in the cancerous cells. Now, you do this by collecting mRNA from the cells, and which are only going to be made by genes that are actually turned on. So if the gene's turned on, it's being transcribed into mRNA, and then it's going to be used to produce protein during translation. All right. Now we can also use this information to determine if there's a correlation between how these genes are expressed and how they interact with each other. All right. All right. I'm going to disappear from this video right now. I'm going to show you about a 3-minute video of how a DNA microarray works, and this is done by an awesome YouTube channel. That's done by a guy that has a great blog. He simply just makes a lot of these HD videos. His name is Dr. Prodigious. So enjoy, and then I'll be back in about 3 minutes.
A microarray is a small silica or glass slide, which is the size of a postage stamp, that contains thousands or even hundreds of thousands of spots. Each spot contains a synthetic piece of DNA with a known sequence. A microarray consisting of partial sequences from a number of specific genes is created by placing a series of known single-stranded DNA sequences in an array on a microscope slide or a silicon chip. The sequences are placed in a specific pattern so that each position on the slide corresponds to a particular known DNA sequence. The portion of a microarray shown here only contains 6 spots, which are labeled A through F. These spots may correspond to DNA sequences found in 6 different genes. The expression of genes depends on the cell type and the environmental conditions. To study gene expression, messenger RNA or mRNA can be prepared from a particular type of cell, such as a muscle cell, or from cells that have been subjected to an environmental agent, such as a hormone. This mRNA that is expressed from such cells is used to make complementary DNA or cDNA. cDNA is made in a test tube by an enzyme called reverse transcriptase. This enzyme makes a DNA strand complementary to RNA molecules used as a template. Special nucleotides that have a fluorescent probe attached to them are used to make this cDNA. This probe will make the cDNA molecules fluoresce green when exposed to ultraviolet light. Once the fluorescent cDNA has been prepared, it is allowed to hybridize with the DNA samples spotted in the microarray. The cDNAs complementary to the type A mRNAs will hybridize with the DNA spot in the array that contains DNA complementary to the type A cDNA. Likewise, the type D cDNA will only bind to the DNA in the spot labeled D, while the type F cDNA will only bind to the spot labeled F. Since the cells did not express any mRNAs that correspond to the DNAs in the spots labeled B, C, and E, no cDNA is made that will bind to these spots. After hybridization, any unbound cDNAs are washed away, and the slide is examined under a fluorescence microscope, where the spots that contain fluorescent cDNAs can be identified. In an actual microarray analysis, a typical cell would express hundreds or thousands of different mRNAs, so that hundreds or thousands of different spots would be identified. Therefore, microarray analysis is a powerful method to study the expression of many genes simultaneously from a particular type of cell or from cells that are exposed to a specific environmental condition.
All right, I like showing you that video because, I could explain it to you, but it's always a lot better to see a video. So that is a perfect description of how a DNA microarray works. All right, so we've done this DNA microarray, and on our little silicon chip, we can see these different colors. So what the heck do these colors mean? Well, if it's red, that means that gene is turned on only in a cancer cell. And if it's green, it's only turned on in a normal cell. If it's yellow, that's mean it's turned on in both. and it probably doesn't have really anything to do with the cancer. If it's black, that means it's not turned on at all. All right, and so it's not turned on in either a normal cell or a cancerous cell, and it probably just doesn't have anything to do with that kind of tissue. So if you remember when we did our DNA microarray, we had that SRY gene in Grandpa Joe. That wasn't turned on in a lung cell because the lung cell doesn't even express that one. So if we were doing a proper DNA microarray, we'd expect that one to be black, as you see here in picture C. Now, one of the things we can get from these DNA microarrays is it depends on how much this gene is turned on. So if you go back to our very first lesson, we were doing the ELISA test, and you had those different colors of blues in your ELISA, that would tell you how much of that antigen would be there. So we kind of get the same thing here in this DNA microarray. The brighter the color means that you have a high expression of that gene. In other words, that cell made a ton of mRNA. If you have a a darker color, that means it didn't produce as much. All right. So as you look over here on this this red series of colors, to the left here, where it's bright red, that was really, really expressed in that cancer cell. If you go a little bit farther to the right, where it's a really darker red, where it's getting near that black, that means it was turned on at a lesser amount. I want you guys to pay particular attention to this slide because it's going to incorporate a little bit more of that math that we were talking about in 314 and in 315. And if you look over here on the left side of your screen, those are those really important bullet points that were in lesson or activity 314. All right, so let's look at these colors and see what they mean. All right, so we got 4 genes here, creatively called gene A, gene B, C, and D. And as you can see, we got the colors of red, yellow, green, some dark near black kind of stuff. All right, so let's look at gene A. All right, when we did a DNA microarray of the tumor cells, it was a bright red, which implies that this was expressed at a high amount within the cancer cells. In a normal cell, it was a very dark green, which means it really wasn't expressed hardly at all. And when you superimpose, or you let the computer When you let the computer superimpose the tumor cell with the normal cell, you got kind of an orange color, which implies this gene's kind of turned on in just a cancer cell only. All right. So if you look down here into the 4th row where it says the ratio of red to green, in other words, tumor to normal, this one is a 4. Okay, 4 times the amount of red as there is in green. Now, what the heck does this mean? Higher levels of gene expression in cancer cells than in normal cells. Therefore, the conclusion is this gene is induced by tumor formation. Think of this as an oncogene. All right. In gene B, we have kind of the same thing. We've got red, full red, almost full red in the tumor cell. It's almost a full green in the normal cell. Superimpose, it's yellow. That gives you a ratio of 0.67. 2/3 amount of red as there is green. So we have a lower expression of cancer cells. So this gene is kind of suppressed. Now in gene C, it's 1, which means there's the same amount in red and green. So this gene's not affected by tumor formation. If you look over here in gene D, we have lower levels of gene expression in the cancer cells than in normal cells, so this gene is suppressed by tumor formation. Okay? So as you look over here, gene A is induced by tumor formation, so think of it as an oncogene, whereas genes B and gene D, those would be tumor suppressor genes, they're turned off, and therefore gene C, since it's 1, same amount in both cells, probably doesn't have anything to do with the cancer. And that's how we can interpret the results from a DNA microarray. All right. So how are the similarities of gene expression patterns going to be shown in different individuals? And this deals with the statistical analysis that was in 315. Now we're going to use correlation coefficients to determine if there's a pattern between what genes are turned on and what genes are turned off. And as you see down here, we've got those bullet points from 315 that you need to know. If there's a positive correlation, they're behaving in the same way. If there's a negative correlation, they're having opposite. In other words, one's turned on, one's turned off. If the correlation is 0, there's absolutely no correlation between them. And the farther you get away from 0, the stronger the correlation is going to be. All right. All right, that's going to conclude this video. Once again, I want to give a shout out to Chad Shelley, fellow PLTW teacher, for creating this PowerPoint. And also shout out to Dr. Prodigious, who created that animation on DNA microarrays. All right, you can find this PowerPoint on My Big Campus if you want to look at it over yourself. So good luck tonight, and good luck on tomorrow's quiz. So, till the next time, we're going to catch you on the flip side.